I will going to share my screen with you guys, and we will going to have our second क्या कहते हैं उसको round of our blended classroom. Here we go. So I am going to share my screen with you guys, and today we will going to talk about the immune receptors and cell signaling. During our earlier classes, I have talked about that how cells communicate with each other. How cells communicate with each other. I have shared with you guys that how cells talk with each other, and that is called as cell signaling. How a cell receives information from outside. It's a complete set of a separate domain of life sciences where you guys should study the concept of cell signaling as a proper subject, or as a proper course at least. It is very fascinating, very important, and a very must-have type of stuff which you should know. Like, you know, there are things which you will have to deal one way or another if you need to stay in life sciences. Cell signaling is one of them. How cells communicate with each other, how cells talk with each other. Whether you are learning developmental biology, whether you are studying differentiation, you are working on differentiation, or whether you are studying immunology, or you are working on our immune system. So in one way or another, you need to know how cells talk and communicate with each other. And that thing is called as cell signaling. It's a separate topic, it's a separate course. But because of the importance of cell signaling, that how cells communicate with each other, and we have talked about certain signaling pathways as well. If you can recall, can you name some of the cell signaling pathways we talked about? शाबाश, मैंने कहीं पर सर्वाइवल पाथवे बोला होगा, शाबाश, लेकिन उसका अपना नाम PI3 AKT pathway है, and that's cool. Okay, that's good. Or शाबाश, or कोई cell signaling pathway which you guys might recall. It's not the cell signaling pathway. My age is distracting me a lot. Or shabash, MAP kinase is cell signaling pathway. Shabash. Koi online Bahadur is trying to answer through the chat. हाँ, G protein के through एक cell signaling हमने पढ़ी थी something. शाबाश. It's a RAS pathway. Khatija has mentioned here. And जो हमने immune receptor से especially पढ़ा था, STAT pathway, STAT, JAK. These were the pathways which we have just heard during our understanding of molecular immunology. That those were required to communicate the information. Those were required to get the information from outside and then to transfer that information to the cells. And then cells started showing the change in the phenotype. If you can remember, I told you that a lot of, I showed you a very weird picture, और मैं वो दोबारा भी दिखाऊंगा अभी आपको. And that picture, like circuits connected within the cells. And out of those circuits, I just specifically mentioned one particular receptor, which is called immune receptor. And that receptor communicates with the gene expression. At the end of the day, that actually trigger the expression of certain genes, and that is how the cell started changing their structure. You guys heard a lot about a cell releases cytokines, the other cell receives the cytokine, and after that they started proliferating. They started increasing in numbers. They started increasing, changing their shape and structure and undergo differentiation. So all these things are only possible if cells receive something, and that something is going to hit certain target genes, and that it can trigger the expression of certain target genes. So we will see that how, in particular in immune system, these signaling molecules work to trigger the expression of target genes. And remember, we are not supposed to learn all the signaling molecules. We are only studying certain ligands, certain signaling molecules which are involved in particularly immune system. And the most famous of them, आप जानते हैं वो कौन सा है? The most famous of them is the cytokine. So we will see what cytokines are, what type of cytokines are out there. We will see how they will going to attach themselves with their receptor, and the name of their receptor is cytokine receptor. And then we will see how the cytokine receptor will forward that information to the downstream genes. And I believe at the end of the lecture we might be able to understand how during an immune reaction cells receives information and respond to those information. So that is the objective. Okay? Shabash.
Yes,
Okay.
sir.
आपको ये दिखाना था जो रह गया ये बतौर खास. You know what are these? These are signals. And this signal काशिफ शायद इसका मतलब लगता है कि लख दी लानत है. But I mean, I don't want to use, but they are the expressions and signals which we use to communicate with each other. But at a cellular levels, cells use a different way. Cell use a physical way using the small physical molecules to communicate information between the cells. For example, these small molecules might be proteins. The small proteins, they might be like glycoproteins. They might be the very commonly they might be the hormones. So those hormones or the small molecules or small proteins, they are capable of binding themselves with the proteins present on the cellular surface. And that trigger a cascade of reactions which we call as cell signaling. And most of the time, they ended up in affecting the expression of certain genes present within the nucleus. Now think that the message was received on the cellular surface. The message was received on the cellular surface and the response was given by the nucleus. So that information on the cellular surface should be transferred to the nucleus with the help of intermediate members, and those are called as messengers, and then they're called as secondary messengers. The nature sometimes amplify those signals in the meanwhile and eventually able to create molecules which can penetrate the nucleus and enters into our nucleus and able to bind on the promoter region of certain target genes. And that is how they're able to trigger the target genes. For example, you guys have heard that in case of cytokines and immune reaction, a cell receives an information from outside and then they start dividing. No, a cell start dividing. That is only possible if the cell started preparing the proteins and the things which are required to execute a cell cycle activity. Those are called as cyclins. The CDKs, those are the molecules which actually push the cells to go from the G1 phase to the S phase, and then from the S phase to the M phase. Okay, and then from the M phase, from the S phase to the G2 phase, from there to the M phase, and then from the G1 phase. So that is how the cell cycle continues. But the cell needs those small molecules, those proteins, which can trigger and push the process of the cell division. That is only possible when a cell receives a signal from outside and then it transfers that information within the nucleus and affects and touch those target genes which are involved in the process of cell division. So activate those genes. This is the cell signaling which is affecting the cell proliferation in the particular example I given to you, and that is called as cell signaling. This is one of the example of cell signaling. For example, if a signal came from outside and it tells the cell, it tells the cells to do what? It tells the cells to stop the cell division. So what will happen in that case? A signal receives on the cellular surface, transfer from the messengers, secondary messengers within the nucleus and touch the target genes which will produce the products which can halt the process of cell division or which slow down the cell cycle, which can stop the cell division. Okay, so that is how it actually talk and communicate with the cells. It's very delicate and very important. So most of the time we receive things from the outside. Most of the time we receive information from a cellular surface. Different types of proteins are involved, and the most famous of them are called as the G protein. G proteins. They are present like on the cell surface. Certain small molecules, they can bind themselves with those proteins. They are called as ligands, and when they bound with the receptor, they actually changes the conformation of the receptor. When a stimulus bind with the receptor, you might ask this question to yourself, that how it is possible that a protein present on the cellular surface, when receive a signal, it started doing something. So that signal, which is captured by the receptor, actually changes the conformation of the receptor protein. It changes the dynamics of that protein and allows that protein to execute newer function. मैं आपको बता देता हूं, भी हम डिटेल से पढ़ेंगे. That newer function is that it allows that protein to start acting as an enzyme. For example, let's make these things a little bit more clear. So this is a cell surface protein. This is the cell membrane, and there is a cell surface protein. This is an extracellular domain, and this is their intracellular domain. Okay, I'm sorry if you're not. So there are extracellular domain, and there is an intracellular domain. When a ligand binds itself with the extracellular domain, it actually changes the conformation of that protein, and the low protein is now able to become doing something new. In easier words, it is called, now that receptor is activated. Now that receptor is able to execute a certain enzymatic activity. Most of the time, that enzymatic activity is phosphorylation. Since you are just at the beginning of cell signaling, so I'm just giving you this one particular example. This is not the law, but most of the time that means the protein or the receptor is now become a kinase, is now become an enzyme which is able to execute the phosphorylation. Now that receptor, which is inactivated earlier, is now able to change one thing, or to change a protein from inactivated form into an activated form, or I must say into a phosphorylated form. It can execute the work. It can attach itself with other things. So that is how the things actually work. And after that, I told you, it eventually ended up in the nucleus. It eventually ended up within the nucleus, where it affects the expression of certain genes, and those genes will start doing their job. Okay, cell signaling is important at a cellular level to control and manage a lot of things. Especially external signals are required by the cells to make decisions. There are certain cells which are supposed to go for the cell division. There are certain cells which are not supposed to go for the cell division at all, like neurons. They do not divide. Skin cells, they are supposed to go for the cell division a lot, because we are rapidly losing the skin cells, so they are required to be replaced. Think about the cells. Think about the cells which are capable of giving a response to estrogen hormone. Estrogen hormone is supposed to produce in the body, and it's a kind of sex hormone produced by the ovary, and it's supposed to affect the function of certain target cells. Now imagine how nature control the cell signaling and presence and absence of receptors to control the function of the cells. The estrogen is present and released inside the body from the ovary. It is all over there in our bloodstream. The estrogen is there, and the estrogen level is increased during a certain period of menstrual cycle as well. And at that time, the blood contains a lot more estrogen than the normal days. If every cell of the body is carrying the estrogen receptor, what will happen? All of those cells of the body will going to bind with the estrogen, and they will respond. I can tell you what is the response. The response of the estrogen is the cell thinks that it requires to go for cell division. So estrogen receptor का cell के ऊपर असर क्या होता है? The cell feels when it receives the estrogen, the cell feels it there is a need to go for the cell division because certain cells are required to produce at that time during the menstrual cycle. So the estrogen helps those cells to go for the cell division. But the estrogen should not touch any other cell which is not involved in that function. If the other cells carry the estrogen receptor, they will respond as well. So what will happen? The nature has terminated the estrogen receptor in all the unrelated cells. So only the cells like breast epithelial cells, they contain the estrogen receptor. Ovaries, certain part of the ovary, they have the estrogen receptor. So only those cells respond to the levels of estrogen in our body. So this is one of the way nature controls that where to send the message. So increasing the level of estrogen in the body overall, but it's not affecting the rest of the cells. So rest of the cells are like same as before. Estrogen do not exist according to them because they don't have the estrogen receptor, and only the related cells will going to respond to the estrogen receptor. So these are the important things with the help of which a cell controls the function, the cell controls the activity. And all those activities are very important for the normal function of the cells. And believe me, if this tightly regulated mechanism is impaired because of so many factors, it can go wrong, and the cell will started receiving the false. I want to talk more on this thing actually. This is the philosophical aspect of cell signaling, that how cell signaling is able to control the same things in a different way, or how they're able to execute their functions differentially in the body. Okay, I will be doing this in the next session. So these are the functions. Let me restart the lecture we have left in the last meeting. These are the things which are coordinated with the help of cell signaling, and you are just looking at a very small part of a very big thing. But cell signaling is like controlling all the functions of life, coordinating between the cells, coordinating between different types of cells. So that is a thing which you should know if you want to stay in life sciences, one way or another. But since we are working only on molecular immunology, so I am just sticking with the cytokine receptor and sticking with the signaling pathway of the cytokine receptor. But this is a way you can guess, or you can understand that the other signaling pathway works in the same way. There will be a different receptor, there will be a different messenger, there will be a different secondary messenger, there will be a different target genes. So that is how it works. So what I wanted to tell you that how the control is actually regulate things in our bodies. So I shared one example with you, and for example, I shared with you guys that there is a cell, and that cell contains a receptor, and that receptor is called as estrogen receptor. Is called as an estrogen receptor. It is present on the cell surface. I am sorry about my drawing. I'm really sorry. And there is an estrogen receptor present on the cellular surface. One estrogen receptor is present on the cellular surface. Suppose it's a suppose value. So if there is an estrogen in the body, that estrogen is a hormone which can bind with this receptor, and you know what will happen? It will send the signal into the nucleus, and the target genes will be expressed, and cell will start doing the function. And what will that function will be? That function will be to go and proliferate and divide. All those cells in the body which are carrying the estrogen receptor, they will respond to estrogen. That's the one best way. The neurons don't have the estrogen receptor, so thankfully they will not respond. Otherwise, the ladies might be periodically mad, you know. Don't feel offended. I'm just cracking a joke, trying to crack a joke, because the estrogen level increases and then decreases in the body. So that means during a particular period of time, you are required to produce more cells in certain specialized parts of your body, not in every body. You don't want your skin cells to respond to the estrogen. You don't want your brain cells to respond to the estrogen. You don't want your other body parts, heart cells and the other muscular cells should respond to the estrogen. You only want the dedicated cells which are part of that system should respond to the estrogen hormone. So once that estrogen is there, the cell will respond and it will go and it will proceed for the cell division and produce more cells. Now think if because of some XYZ reason, if the estrogen receptors present on the cellular surface, if the number of estrogen receptors present on the cell surface are increased. I just shared a suppose example that typically if a one estrogen receptor is present per cell, how about now having 4 estrogen receptors present on each cell? So what will happen in this case? If we have an estrogen over there in the blood level, now think about the two cells. Compare these two cells. Compare these two cells. Which of the cell will be more sensitive for the estrogen? The answer is this one, which has the 4 estrogen receptor. This is like 4 times more sensitive for the estrogen. It will rush or respond to the estrogen 4 times more than the other one, the normal one. So if the level of estrogen increase in the body, the cell which is carrying more receptors will going to respond faster as well. That will going to produce more cells, and those more cells will have more estrogen receptors. They will become more sensitive, and then they will going to produce. The daughter cells will have the 4 estrogen receptor. The daughter cells will have the 4 estrogen receptor, and then the process continues, and cell will grow and then start producing a ball of cells, a mass of cells, which is called as the tumor. And that happens in most cases of breast cancer. The number of estrogen receptor increases. When the sample was sent for the very first time out of the mammography, when they scan the body and find a weird sort of mass, they use a needle to take out certain cells from that mass using ultrasound machine, so that they able to insert the needle right inside that weird looking mass, which they were suspecting at that time that it might be a tumor. This process is called as biopsy. When they pick the cells in the needle, they send their cells to the pathologist, laboratory में test canonically based. They analyze different things, and one of the things they analyze is that they write in their answer that this is an estrogen positive, ER positive breast cancer. You can recall this term अगर आपने जिंदगी में खुदा ना खास्ता कहीं से सुनी है, लेकिन आप researcher and students are, you should know. So they write it's an estrogen ER positive breast cancer. So what that ER positive breast cancer means to you now, that it is a kind of cancer right now which you are looking at, which has more estrogen receptor than normal. So that is why it converted into a tumor. So there are 3 basic receptor markers which they usually analyze: the estrogen receptor, the progesterone receptor, PR receptor, and the third is the HER2 receptor, the human epidermal growth factor receptor. So in totality, they usually answer like ER positive, PR positive, HER2 positive. But if it is written ER negative in the report, now you understand if it was written like ER positive in the report. What that means? The person, the unfortunate person, developed breast cancer because of the overproduction of estrogen receptor. Can you deal with this type of cancer? Can you deal with this? This type of cancer can be dealt easily because we have a molecule called tamoxifen, and that molecule mimic the estrogen. So we have the tamoxifen molecule which can mimic the estrogen, and it can bind the estrogen receptor and block them. So the cell division, the process of the cell division and the response of the cell decreases, and with this therapy, the tumor size will start decreasing over the period of time. Estrogen correspond करना ना, so all the receptor will capture the tamoxifen. So tamoxifen will bind with the receptor, but they will not going to induce the cell signaling. So it is like they occupy the receptors. This therapy is called as hormone therapy. And if a person is diagnosed with ER positive, a PR positive, a HER2 positive sort of breast cancer, the doctors were somehow hopeful and they believe that they will be able to deal with this comfortably because they do have a drug which can go and bind with the target receptors. It makes a lot of sense. At least the tumor will not increase. And tumor, the presence of tumor is not the cause of death, I can tell you. The presence of tumor and their potential, their ability to start proliferation, that is the reason of death. So if you can contain the tumor, you can deal with the things comfortably. You can remove it by surgically and by, but the good news is that the tumor will not grow beyond that thing. It is like, let me, the time is not up. It is like, we have a reason of the breast cancer. That's a good thing. If you know the thing, if you know the reason, you can create, you can have a drug and you can use it for that purpose. It is like, अगर आपके छोटा भाई है, छोटा भाई वैसे ऐसे होते हैं कि एक छोटा भाई है जिसे गुस्सा आता है जब भी, he go to the kitchen and broke a glass तोड़ता था. मतलब ये उसका स्टाइल है. उससे में जब भी आपको किचन में एक टूटा हुआ ग्लास मिलता है, you can predict कि ओहो छोटे भाई को गुस्सा आया होगा किसी बात पे. You have a reason, you have a response, and you can make corrections, you can deal with that thing, and at least you know the trouble. आप उसे गुस्सा नहीं चढ़ाएंगे बस आइंदा से तो ग्लास ही टूटेंगे. It is as simple as that. Think about if you start finding the broken glass in your kitchen और भाई को गुस्सा भी ना आया हो. हमें तो गुस्सा भी नहीं आ रहा आजकल, वो कह रहा है मुझे कोई गुस्सा नहीं आ रहा, I'm fine, and you still like finding broken glass in your kitchen. It's a weird situation. It's something which you don't know how to deal. पहले क्या था, भाई को डील कर लेंगे, we will make him happy, glass टूटेंगे. You know the solution, but in this case you don't have the reason. Now think if the pathologist answered the report. The pathologist said, Janabi Ali, this is a breast cancer, the ER negative. What that means? The estrogen receptor are not overexpressed, but still the cells are growing. So that means now we don't know. The estrogen receptor is not the reason. Now the doctor will going to give the tamoxifen to that patient. It will not work. So now the doctor will start it, oh, it's the negative, ER negative, or PR negative, or even the HER2 negative. This is called triple negative version of cancer, and this is the most advanced, rapidly proliferating form of breast cancer. And then the doctor was like, "Khatre wali baat hai. It's like triple negative. We don't know how to contain that thing. We have no reason, obvious reason. We can try to remove the tumor. We can give the radiation. We can do the chemotherapy. But we don't know how to stop the tumor. If some of the cells were there, they will keep on growing. And that is the big trouble. And all these things are like because of the relation with the trouble in the cell signaling. So at the end of the day, you will see that how cells communicate and talk with each other. That is the primary mode of regulation between the cells. And that is the way cells can live together comfortably hereafter. So sab hasi khushi se rehne lage. That depends that they should communicate with each other normally, and that is how we can grow our society in our classroom and our friendship circle as well. You guys communicate with each other, you understand each other, you know when to speak, when not to speak. You know ab se bada gussa aa raha hai, isse aage karenge tap jayega ye. You need to stop the conversation at this time. So cells should understand all these, should be able to respond to all those signals. And if you understand that how the cells respond at a cellular level, actually that is an indirect way of understanding how we react to the external signals. In the broader concept, in the broader perspective, if you understand that, you know, the cells are, आपने एक बचपन में एक बात सुनी होगी बायोलॉजी में, खुलिया जो होता है वो cells के structure और उसके function की इकाई होता है. It's like a structural unit of cell of life. That means every life is made up of cells. Easy. The second part is that cells are functional unit of life. That means whatever we are doing as a life form, we are doing all these things because our cells are doing all this thing. My online class might be thinking they are alien. I'm not looking at them. They feel, you know, kya kehte hain usko? Alienated and left out. Okay, guys, don't worry. So the point is that, क्या मैं बता रहा था? हाँ, so in terms of cell signaling, whatever the signaling things we are doing at a molecular level, at a cellular level, that is actually is a way that how we communicate with our external signals. आज मौसम अच्छा है, okay, that is an answer. There is something related to that feeling at a molecular level. आज बहुत गर्मी पड़ी है. What are the things we need to do? Whatever we are doing, we are actually doing all these things after that in order to communicate between the cells normally. तो ये बहुत important factor है. इसको relate करना मुश्किल है जो मैं कह रहा हूँ आपसे. जैसे भूख लगी है, खाना खाना है. At a molecular level, it has a different meaning. Okay, so let me conclude my class here today, and we will be able to learn more things in the next class. But I want to take your questions. If you are, I just stopped this thing here because I want you guys to talk for the next 10 minutes. It's your time. Show time. G. Abnormal receptor. When you said के बाद वाली जो statement है वो बहुत important होती है. You know, I said the number of estrogen receptor increased. Okay, the number of, like earlier it was like 1 receptor per cell. Now there are 4 receptors per cell. It's like increase in the number of. That is because of the overexpression of the receptors. Okay, so we have a cell with more receptors. All are same, doing the same things. No, but the cell is able to respond to the estrogen more than earlier because it has 4 receptors. Now the tamoxifen has a structural similarity with the estrogen, but because it binds with the pocket of the estrogen, but since it is not the estrogen, so it will not able to change the conformation of the receptor. It will not going to change the dynamics of the receptor. It will not going to activate the receptor. So it will bind and irreversibly block the estrogen receptor. All the estrogen receptors. So normal cell नहीं काम करेगा ना. That's a risk which it's a good point. आपका नाम क्या है? हमने ने पूछा कि सर अगर हम tamoxifen use करेंगे, that will going to block all the estrogen receptors. So that is not nice. I mean, we do need the function of estrogen, and the answer is yes. It is not nice. Cancer is even worse than nice. Only facing something which is not nice versus something which is life-threatening. So we choose a calculated risk at that time. Actually, doctor defines the dose. वो ना इसको define कर सकते हैं कि वो आपसे सवाल पूछते हैं, वो पेशेंट से क्वेश्चंस करते हैं और उसके responses और उसके answers पे define करते हैं कि how much tamoxifen I should give that patient, which actually allows that patient to execute some of her own normal function as well. तो वो कुछ ना कुछ मार्जिन रखते हैं इस बात का. It's a very good question. ये डॉक्टर की जहानत है ना. अगर वो ओवरडोज कर देगा तो सारे एस्ट्रोजन रिसेप्टर ब्लॉक हो जाएंगे. एक्चुअली वो लॉट ऑफ थिंग्स वर देयर, आई कैन टेल यू. आई मीन वो देख रहा होता है कि ये लेडी की एज कितनी है, ये मेनोपॉज हो चुका है नहीं हो चुका. आई मीन she is a mom, a breastfeeding mom, or a lot of things the doctor is actually looking at, and there are a lot of functions which are related to the estrogen. So in that case things will be more complicated, but most of the time the breast cancer patients, the ladies, they're like over 45, 50, 60. So most of the time they can sacrifice. They are like ready to compromise their functions of ovaries. वो तैयार होते हैं कि it is it's not a very difficult risk to take at that age, but in case of a younger lady it's quite difficult, because you can guess they will not going to able to use their ovaries in a normal way during that treatment and after that. And during the chemotherapy, I can tell you that it's almost destroyed. During the chemotherapy, the functions of ovaries are almost like gone. It is very difficult for them to have a baby after that. Gee. ये मैंने आंसर किया ना. Doctor is the one who is taking all these factors into consideration. उसका experience है ना. Actually, experience is this thing. There is more. Experience is very special because with the help of experience, we are able to, the doctor is able to understand ना कि इस तरह के मैंने 50 पेशेंट देखे थे, उनकी इसी तरह की symptoms थी, and I recommended that much amount of dose to these guys, and that is how they learn कि कितना चाहिए, वो symptoms की बुनियाद के ऊपर, age की बुनियाद के ऊपर, इन चीजों की बुनियाद पे, nature of the tumor, size of the tumor, इन सब चीजों की बुनियाद के ऊपर फैसला करते हैं. This is not the cancer biology class, so let's move to the immune system. So the purpose, the reason I wanted to show the earlier slide to you guys was that okay, you should know कि nature के पास एक जबरदस्त तरीका है communicate करने का. तरीका नंबर 1, ये जरूरी बात है. तरीका नंबर 1, nature communicate करती है with the help of a chemical, hormone or a protein. This is one way. Producing a hormone, getting the response, one way. The other way is that having the receptor in the target cells, producing the hormone and then giving the receptors to the target cells. So the target cells only have the receptor, they will going to respond only. Not every cell is required to respond to this. ये जो delicate किस्म की communication है, I'm sorry for this difficult things, ये development को control करती है. During the process of development, that thing is required. We have a ball of cells. Certain cells are transforming themselves into ectoderm. Certain cells are transforming themselves into mesoderm. The other cells are transforming themselves into endoderm. All these things are required a coordinated effort or communication between each other. So if a chemical is produced and it should only affect the ectoderm or ectodermal cells, it should not affect the mesodermal cells or it should not affect the endodermal cells. So क्या nature कैसे control करेगी? Nature do not give that receptor of that chemical to the mesodermal as well as to the endodermal cells. उनको वो receptor नहीं देगी, so they will not respond. Okay, that's a cool thing. आप लोग एक सवाल में हमने पहले डिस्कस किया होगा शायद. I will ask a question to you guys before anybody. Somebody wants to talk, please speak. जी. तो विलन की बातें सोचता है, विलन के दिमाग से सोचता है. काशिफ कह रहा है बायो वारफेयर में क्या होगा. हाँ, मैंने आपको लास्ट टाइम एक बात बताई थी ना, टी सेल्स इंजीनियर करना, आप लोगों के साथ बात करते हुए, वो बड़ी मजे की बात थी, लेकिन वो गलत बात है. वो इट इज लाइक, आई मीन एज ए रिसर्चर यू शुड नो अबाउट ऑल दिस थिंग्स. टी सेल्स क्या है, सीटीएल्स क्या है? सीटीएल्स सेल किलर्स हैं, एंड दे वर ट्रेन्ड अगेंस्ट a particular antigen. They will go into the cell freely, frankly. If I ask you, the CTLs are passing through the brain cells में से गुजर रही हैं वो? Yes. Okay, blood reaches everywhere within our body. If I ask you, the CTLs are passing through the heart, the answer is yes. Passing through the lungs, the yes. Skin, yes. They are passing through everywhere. But those CTLs are not killing those cells. If that CTL are against hepatitis C virus, for example, those CTLs are passing through the kidneys. Nephron में से गुजर रहे होंगे ना, गुजर, but they are not doing anything to all the cells in the nephron. But when those CTLs pass to the liver, जहाँ पर infected cells हैं hepatitis से, those CTLs will start killing those cells. So that is how nature control in the same way. So CTLs are there, but they're like non-functional. So every other cell is invisible for them, other than that one cell which is carrying the information. वो जो antigen के against, जिस antigen के against CTL raise हुआ है, if the cell is carrying that antigen, the CTL will go, bind and kill those cells. अब अगर आप इस CTL को engineer करें, particularly against certain cells and receptors which are present in Kashif. So those CTLs, if they are transfused, like if they able to reach to everyone's body, they will not going to affect them. It's an invisible thing but those one particular cell that are present in your body will going to be the target. But think like a hero. Think like a hero. Creating, मैं conclude कर रहा हूँ, ये काम की बात है, ठीक है, ये ले जाना, ये आज का immunology का तवीज़ है. Engineering cytotoxic T cells in such a way that it should be able to recognize the cancer cells. Getting the CTLs or finding the CTLs which know the cancer cells, and then multiply them, help them, grow them, increase their number, and then put back into the person's body. Earlier, the person is carrying like few CTLs. Now the person is carrying like thousands of CTLs. When those CTLs are trained against what? Against the cancer cells. They will go, they will not harm any cell of the body. They will only kill selectively to the cancer cells. लेकिन उस वक्त में उस स्टेट में जहां पर उस इंडिविजुअल का CTL आपने लिया, that is perfect for that person at that time. वो तो अगर उसको आप अलाउ करेंगे तो कुछ अरसे के बाद म्यूटेंट कर जाएगा ना. But that drug, देख लिहाज से ड्रग बन गया ना. It is called, this process is called as cancer immunotherapy. It's called cancer immunotherapy. It's very important. इसका इलाज, इसकी मदद से हमने एक इलाज कर चुके हैं. अब गूगल कीजिएगा, एक रिपोर्ट है, हम इसकी वजह से हम एक पेशेंट को ट्रीट कर चुके हैं. It's already like applied in human treatment. उसको मारते हैं. जब तक वो infected cells उसको lose नहीं करता है. The question was that if the PD-1 receptor is there in all the cells, how it is possible that the effector cells or CTLs will be able to kill that cell as long as the PD-1 domain is there? The answer is yes. As long as the PD-1 domain is there, the CTL will not be able to kill that cell. When the cell become cancerous, यार ये मुझे इधर ना लेके जाओ, मैं खत्म कर रहा हूँ बस. If the cell become cancerous, you know what will happen? Certain checkpoints are passed. टाइम नहीं है लेकिन ये बड़ी मजे की स्टोरी है. Certain checkpoints were not regulated. Before the cell go for the cell division, I can tell you, the cells have made lot of calculations, regulations, and they made lot of decisions. Okay, the cells, before they go for the cell division, they analyze certain things, whether the food is enough, whether there is a need to go for the cell division, and whether the genetical structure is perfectly intact, no toxins are there, everything is fine. And this process is done with the help of certain important proteins, markers, and molecules. Those are called as cell cycle regulators. If those things are there, cells will think, I can move from the G0 phase or the G1 phase to the late G1 phase, and then the cells enter into the S phase. You know S phase क्या होती है? When the cell started duplicating its DNA. Before the cell jump into the S phase, it checks certain things, presence and absence of certain things. If everything is fine, the cells commit itself to go for the S phase. S phase के बारे में याद रखें. If it is started, you cannot halt the S phase right in the middle. ये कैसे हो सकता है ना? आप आधा DNA बना लें, आधा रहने देते हैं, आधा क्रोमोसोम डुप्लीकेट कर लेते हैं, आधा नहीं करते. Once the replication started, it should be completed. So before the S phase, the cells check everything and then make a commitment and then go for the S phase. All those things are, and during the S phase, they also analyze whether the DNA is created perfectly, there is no mutation, there are no errors. If the errors are there, it should be removed, all these things. Because if the cell stopped making DNA during the S phase, what will happen? क्या होगा बोला? Cell will be dead. उस वक्त सेल रुक नहीं सकता है. ये बड़ी अजीबोगरीब स्टेट है. आप सेल के बारे में सोचें, अगर एक सेल ने कमिट कर लिया है एस फेज को, इट स्टार्टेड, उसने डीएनए को सिंगल स्ट्रैंडेड में कन्वर्ट किया, स्टार्टेड मेकिंग न्यू बेस पेयर्स, इट शुड स्टार्ट एंड फिनिश द डुप्लीकेशन ऑफ क्रोमोसोम. दरमियान में नहीं रुक सकता. अदरवाइज क्या होगा ना, एक सेल है जिसमें which have 1.5x of our chromosome, that will ended up in like the cells which has different numbers of chromosome, and it never happens. हमारे पास 46 क्रोमोसोम है, 46 क्रोमोसोम होते हैं, कभी 46 पॉइंट या 40, 57, 55, 58 क्रोमोसोम्स नहीं बनते, because if the process is halted in the middle, क्या होगा? Cell will be dead. The same things are applied to the other parts of the cell cycle as well, but मैंने आपको ये बताना है, the cells control that things, check and then proceed with the. In case of cancer cells, what's going on? Lot of pressure is there because of external and internal reasons to go for the cell division. Certain checkpoints of the cell cycles, they got impaired. वो काम नहीं करते हैं. So what will happen? वो cell को check किए बगैर ही push कर देते हैं to go for the cell division. Before checking that whether the need of the cell division is there or not, they push the cell to go for the cell division. Before checking whether there is enough food or not, they push the cells to go for the cell division. Before checking whether the DNA is created perfectly or not, they push the cells to go for the cell division. So what will happen? The daughter cells will going to accumulate more troubles. They will be different from the parent cells. Not suddenly very different, they will be a little bit different. But if the process continues, there will be more errors in the daughter cells, there will be more errors in the daughter cells, there will be more errors in the daughter cells, and that is why after a certain period of time, the daughter cells will be very, very different from the cells they were created, original cells. It's both मुख्तलिफ होते हैं. And that is the reason behind the ever-changing dynamics of cancer cells. The accumulation of mutations over the period of time, and that actually changes what? That changes the cell structures, that changes the cellular receptor, and that can also changes all the good signs present in the cell, including the PD-1. And that is the time when our immune cell will started understanding, oh my God, this is something, a very bad thing. When it lost all the good signs and sequences. Before that thing, it's like immune system put a blind eye to all those activities of the cancer cells at the initial stage. But now, it's bad. And then they started doing their job. Hope this answers the question. The cancer story is lovely. I will tell you this story, the cancer story someday about the cancer cells. मैं वो किसी कहानी सुनाऊंगा. और आपका मेरा वादा, you will fall in love with the cancer cell. Okay, guys.